Hello everyone, I'm Kyowon Jeong, a postdoc in Kolbach Lab in University of Tübingen in Germany. In this poster, I present FLASHIda, an intelligent data acquisition for top-down proteomics that doubles proteoform identification count. This work is also orally presented in this ISMB CompMS course track, so please go ahead and check the video for more detailed information. In this work, we try to tackle the issue of data acquisition in top-down proteomics. Since data-dependent acquisition for complex protein sample analysis has been optimized mainly for bottom-up proteomics, the acquisition for top-down proteomics has much room for improvement. But complex signal structure of proteoform ion hindered the development of a proper acquisition. One important and challenging step to disentangle complex proteoform signal is the mass deconvolution, in which scattered peaks from a single proteoform are converted into a single peak representing the monoisotopic mass of the proteoform. Recently, we have developed an ultra-fast deconvolution method, FLASHIda, that achieves millisecond order of running time per spectrum. By leveraging its fast runtime and accurate mass deconvolution, here we developed FLASHIda that enables selection of high-quality precursors of diverse proteoforms. FLASHIda is an acquisition method, so it runs inside the MS instrument. Currently, it runs on thermoscientific IAPI computable machines. The block diagram in the left side shows how FLASHIda works. FLASHIda takes the MS1 spectra real-time from thermo IAPI. Then it processes the spectrum to select precursors and send the selected precursors to the instrument back again via thermo IAPI for the next cycle. On receiving MS1 spectrum, FLASHIda converts it to mass quality spectrum. MZ to mass conversion is done by FLASHIda, and intensity to quality is done by Q-score calculator. On the mass Q-score spectrum, top and Q-scoring mass are selected for acquisition, interfacing with the quality-based mass exclusion list. The middle top panel shows the Q-score calculator. Q-score is the probability that each precursor is identified, estimated by a logistic regression with selected 6 features from raw signal. The middle panel shows the features. So for example, assume we want to measure Q-score of the green precursor envelope. Note that because this measurement is done after mass deconvolution, we have distinction between signal peaks and others given a mass. The green signal of interest is first compared against the theoretical averaging isotope pattern. Their cosine similarity becomes the first feature. And since we know theoretical signal, we also can calculate noise component and in turn signal to noise ratio, which is the second feature. And the green signal intensity is compared against the signals of different charges from the same mass, in here shown as blue and purple color coded peaks. Their ratio is also considered as the third feature. And these signals of different colors are summed in the mass dimension, and the cosine similarity and signal to noise ratio for this summed signal are added as 2 additional features. And lastly, the average ppm error from theoretical masses are taken as the last feature. We used more than 10,000 spectra to train the model, and the resulting ROC area from 10-fold cross-validation exceeded 0.9. The bottom side of the middle panel shows how the masses are selected on the mass Q-score spectrum. When we use top and Q-scoring selection in this transformed spectrum, selection of high-quality distinct masses is very easy. But if we select top and intensity peaks in raw spectrum, as in bottom-up proteomics cases, in many cases we select peaks from the same mass. On the right side box, the quality-based exclusion is illustrated. FLASHIda keeps total Q-score, or TQ-score, for each mass acquired over a short retention time duration. TQ score over mass is the probability at least one of MS2 spectra from the mass is identified, or equally, the probability that the proteoform of the mass is identified. If it has been identified already, it should be excluded from the acquisition, so any masses of high TQ scores are excluded from the acquisition for a given time. To benchmark FLASHIda, E. coli lysate single shots were used. FLASHIda was compared against standard acquisition with two different retention time gradients, 30 and 9 minutes. The bar graph for proteoform count shows that FLASHIda almost doubles the unique proteoform count for both gradients. And also it shows that 30-minute gradient with FLASHIda is comparable with 9-minute gradient with standard acquisition. In terms of number of proteins, it showed over 30% increase over the standard. The bottom graph shows the number of MS1 spectra with distinct precursor masses. Since we used top 4 acquisition, n is from 1 to 4. While most of MS1 spectra from FLASHIda resulted in 4 distinct masses, the standard is shown to select many redundant masses. This bar graph also shows the portion of identified and unidentified spectra. Light-colored portion is the unidentified portion. By comparing light and dense colored portions, we can see that the identification rate from FLASHIda is much higher than from standard. In summary, FLASHIda enables selection of high-quality precursors of distinct proteoforms without major changes in the experimental setups. And you expect more IDA methods could be developed for different proteoform resolution studies, such as targeted deep proteoform characterization. Thank you.